So in the last video, we discussed the law of segregation and how different alleles from each parent separate during meiosis when cells go from diploid to haploid when the homologous pairs split, and then each individual offspring gets one allele from every single parent. And so now we're going to take a look at Punnett squares. And these will help us discuss what we call random fertilization. Which basically means that it is entirely random, of course, not only which allele goes with which sperm or which egg, but then therefore when two parents are trying to produce an offspring, which sperm happens to meet up and fertilize which egg, right? Of all the possible sperm that a man could make, or a male organism, and all of the possible eggs that a female organism could make. On top of that, all of the possible options of which ones could happen to actually come together to make a child, that is random fertilization, okay? And again, this goes back all the way to like the pros of sexual reproduction is all of this possible genetic variation and diversity. So anyway, how can we predict the likelihood of one random fertilization happening over another? We can use a Punnett square. The most basic of Punnett squares is a square box, but it looks like a window. So it has 4 boxes like that. And let's say we were crossing, let's say, just for example, that a dominant color for flower petals was red, and a recessive color was white. Let's say I were to cross 2 individuals. I'll write the cross on the bottom here. Let's say I cross a homozygous red individual with a recessive white individual. And some people care which side of the boxes it goes on. There are 2 conventional sides, the left side and the top side. You have to use those sides, but it doesn't matter for us which parent goes where. So I'm just going to, I usually start with the top. I'll put the 2 capital R's. So that is our homozygous dominant individual. And I will put the 2 lowercase r's, I'll do it in orange. So those are our 2 parents. This is parent number 1, and this is parent number 2. Parent number 1, parent number 2. Okay? And because I've separated them, you can sort of imagine these lines extending through these alleles, right? So the alleles have separated, and we just follow through sort of down and across, all right? And so I'll erase those. But so if I follow the capital R's from parent number 1 down, this has a capital R, and this one has a capital R. Following down this way, this one has a capital R, this one has a capital R. Then I can go across, right? This one has a lowercase r, this one has a lowercase r, this one has a lowercase r, this one has a lowercase r. And then you will be asked about the likelihood of certain offspring being certain things. As you can tell, these are all the potential offspring we can get from this combination of parents. You can tell it is a 100% likelihood of having a heterozygous individual. So that would be if I asked you the genotype. If I asked you the phenotype, you would say there was a 100% chance of having a red flowered offspring.